So before the break, we want to at least tee up the second big topic.
Yeah. So our second session yesterday was about real world treatment, and the two sort of overarching questions were using real world treatment for valid inference. When is it that we can look at treatments delivered in the real world and have valid inference about the effectiveness and safety of those treatments, and then when people are getting treated in real world settings, how can we appropriately monitor whether they're being treated safely and respond appropriately to potential adverse effects of treatments? So you can see here the sort of beginning of there, and we can sort of scroll down. Once again in the sort of red are some of the points that came up yesterday. The idea that when we're looking at adverse events or potential toxicity of treatments, some of those may be less likely to be specific to a particular indication than the effectiveness of the treatment. A really important point that came up over and over again, that our questions about safety monitoring are very different at different stages of a product life cycle. When a product is new, it's really a sort of discovery process of saying, how could monitoring the use of this, help us to discover a problem we don't know about? While when a product is well established, it may be we know pretty much about what the problems are, but are we keeping people safe when those problems happen? So there's an inference task that's more important early on, and there's more just a kind of doing the right thing by people task that may be important later. That's that point about the sort of conflict of interest. And then the point about, this point about will the trial change the understanding? We talked about, especially, Rob made the point about monitoring for non-serious adverse events, and I guess potentially even for serious adverse events. If a product has been around for quite a while, we have to ask the question, will this tell us something new? In this trial, are we really going to discover a new adverse effect that we did not know about before, especially if the size of a new study is relatively small compared to the historical experience? Then some of the other points that where the research is performed affects the results. Some of you may recall the discussion about, this came up in the sort of warfarin versus NOACs example, but some of the other examples we talked about, that when we're talking about treatments that may be more what I would call operator dependent, by that I mean, the treatment could be well or poorly applied and there's more variability, versus a treatment that would be less operator dependent, or to put it bluntly, idiot proof. An idiot proof treatment might have pretty similar effectiveness and safety in all settings, while an operator dependent treatment might be highly variable. And so the comparison of an idiot-proof treatment and an operator-dependent treatment might yield very different results in different settings. And that's important to bear in mind, both to know that that is the case and to know that there is, I don't know how to say it, potential finagling that could go on if we're choosing our setting to deliver the desired result in that comparison. This sort of selection of the setting also led to some discussion about the sort of enriched designs, low risk versus high risk. Enrichment designs that enroll more higher risk people may be more likely to demonstrate benefit, but also may raise more concerns about people who are at higher risk being exposed to new treatments. So the assay sensitivity and some of the concerns about safety. The quest, I think, the point Sebastian made about the difference between the study. I think you talked about, are we studying a molecule or are we studying a care pathway? That it gets when a molecule may have similar biological effects across settings, but if we're interested in how molecules interact with care pathways, that may be setting specific. The point about collecting data on non-serious adverse events may not be helpful. Then skipping down when we talked about questions about sort of adherence or how people behave, because the question about how people behave not just has to do with their adherence to medications, but other behaviors which may affect their health states. To me, the shining example that came up from that was the pre-exposure prophylaxis, for prevention of HIV transmission, where if we were to enroll only people who would always practice safe sex, we would never demonstrate a benefit of pre-exposure prophylaxis because the outcome we fear would never occur. So we must enroll real-world people who behave as real-world human beings would do. A discussion about blinding. The idea that, we certainly always want to try to blind outcome assessment, ideally blind analysis. Whether we blind patients and providers to treatment, the answer is it depends, but I think we were able to get to it depends on what. That blinding does constrain treatments to be the same, which may sometimes be okay and sometimes not okay. But it may be important to blind patients and providers if unblinding them would somehow bias the ascertainment of outcomes, or the point Bob made, if it might lead people to get different treatments or the treatments to be applied with more or less intensity if people are unblinded. Go ahead and keep going down there. That we talked about, I skipped over that one. We brought up the impact AFib as an example of what I would call a nudge trial. The idea that can we make randomization much easier to do by what we're randomizing is a nudge to use a treatment more often rather than a 100% likelihood of using the treatment, and that may allow us to draw valid inference. As some of you know, impact AFib will randomize 75,000 people optimally. So even if there is a relatively modest signal, the sample size might be large enough to detect the downstream effect of that signal. Then you can keep scrolling down there. Okay. Yeah. Okay, so we're done with that one. Okay, thanks. So those are the things that came up in the second session. I guess do we want to go to break, or do we want to start doing the discussion of that? Mark, what do you think?
Probably a good time to break. I'm not sure we'll have a chance to get into too much detail. We've been going for an hour and a half already.
Okay. All right. Okay.
And maybe reconvene at 10:45.
Yeah.
All right.
Okay, we'll do that.